Thanks, Anushka. If I'm too loud, tell me. Hopefully it's not too loud back there. Okay. So what I thought I'd do in the next 40 minutes or so is tell you about scientific discovery, which we stumbled upon serendipitously. I'll just dive in. Hold on, let me see. Yeah. There is a reason why I had titled it Monoamines, Mitochondria, and Mood. It's 3Ms. 3M is a company that is known for innovation. And one of the biggest pieces of innovation they had are these post-it notes. But if you know the real story behind it, it's quite interesting. They tried to make adhesive for airliners, and they ended up making a terrible adhesive that did not stick well. In fact, it did not stick well. Under pressure, it stuck, and then it just fell off. So for the longest time, this was a failure, right? It was an absolute failed experiment until someone came up with the idea, actually someone in the company came up with the idea that this would be a great way of tacking on notes, which you wanted to remove at some point in time. When you think of 3M, you often think about their breakthrough product, and their breakthrough product is the Post-it. In fact, the Post-it has become synonymous in some ways with 3M technology. The reason I'm bringing this up is because it started with a failed experiment. It started with something that went exactly opposite to what they were predicting. I want to tell you the story about something that we stumbled upon, which also started as not something we anticipated at all. We were doing experiments looking at the potentially neuroprotective effects of plant-derived. So we received a large grant for that, and we used a bunch of interesting Indian traditional medicinal plants, which the company that had funded us was interested in our looking at, and we looked at them and we managed to kill neurons very successfully. Rather than having anything protected, we ended up killing most neurons in the dish. Along the way, we began to realize that if we better have a positive control, which shows that we are actually know what we are doing, and that it's not that we just kill neurons in a dish routinely, but that we can also make neurons look healthy and look like they are surviving. So obviously a natural substance to put in is to put in growth factor. And so we use BDNF as our positive control. And yes, thankfully our experimental hands were okay. BDNF, as expected, made the neurons live really well. But my lab has been interested in monoamines forever and has been interested in serotonin in particular for a very long time. So if you tell people to start spritzing anything and everything on neurons, one of the things that inevitably comes into one of those dishes is serotonin. And one of our readouts was actually to look at mitochondria because they are a good measure of neuronal survival and neuronal protection. In the process, we stumbled on a discovery that monoamines, both serotonin and norepinephrine, are potent regulators of mitochondria. This was a little unexpected. It was not what we had originally anticipated. If anything, it was because of a failed experiment, which we eventually just didn't find anything very interesting with the medicinal plants in this particular design, but we ended up stumbling on something really interesting with serotonin and norepinephrine. That's why I'm using the Post-it note, as it can change your entire trajectory almost for the entire lab group. Okay, so let me tell you about what we stumbled upon. But before I do that, I want to tell you, for this audience, it's very interesting. When you think about serotonin, in popular parlance, when you say, are there neurotransmitters that are associated with mood regulation, the few that will immediately come at the top of the list will be the monoamines. Serotonin, norepinephrine, dopamine will be right there at the top of the list of neurotransmitters that influence mood. But when you think about it from an evolutionary perspective, they predate the existence of the nervous system. So if you look at the earliest stages of metazoan evolution, monoamines are there long before anything even remotely resembling a neuron shows up. So they are old. If anything, they got co-opted to become neurotransmitters. So for a very long time in evolutionary history, they had functions long before they were actually doing things like signal transduction between neurons, or signal communication between neurons. Oh, okay, sure. So if you think about what the sort of evolutionary timelines for serotonin are, it's 3 billion years ago. So it shows up at the earliest stages of metazoan evolution. It's abundantly found in plants, which we perhaps don't pay as much attention to. There's more serotonin in a walnut than there is in your brain, but you're not possibly necessarily thinking about that. There is a ton of serotonin in plants, and it's just a body of literature that neuropsychopharmacologists, neuroscientists, psychiatrists sometimes have just forgotten about because it hasn't shown up in their standard sort of thing of things that they're thinking about. So there's much, much more serotonin in plants than we appreciate. And actually, after we made the discovery by chance and fluke, we went back and looked at literature. And that's when I began to realize that I, like many other neurobiologists, had also ignored all the body of literature that exists for serotonin in plants. So just sense, neurons are only going to kick in something like 600 million years ago. So there's a very long period of evolutionary time in which serotonin was doing things in cells and in living tissues, which was not associated with anything to do with neurons. In fact, there's much more serotonin, for example, than your gut than there is in your nervous system. Receptors, as we understand them, GPCR-coupled receptors, which is what most of the monoamines signal to, with the exception of the 5-HT3 receptor, all of those also show up somewhere around 760 million years ago in terms of evolutionary timelines and history. So you had monoamines, but they were not doing receptor-mediated signaling for a very, very long time in evolution. So this is interesting to think about. I found to put forth some of the ideas of what serotonin might have been doing in its pre-nervous roles, before it became a classical neurotransmitter as we think about it, a modulatory neurotransmitter. So there's been a lot of evidence that suggests that serotonin works almost as a trophic factor, like a growth factor. There's very elegant evidence that it works like an antioxidant. And there's some beautiful old papers in which people have looked at the browning of leaves. So if you chop off a leaf, the part that you chop off is going to go brown. And that's pretty standard. If you now apply serotonin to those leaves, the rate at which the leaf browns is slowed down. Now, that's an indicator of potentially antioxidant-like effects of serotonin. And this opens up very interesting ideas as to what serotonin might have been doing in other systems. In primitive metazoans like Prenervous, it's a direct regulator of regeneration. Did I just lose my slide? Probably did. Oh, it's come back. Okay. It regulates RNA synthesis. Very recent, very exciting work coming from Ian Mays's lab. So serotonin also works like a post-translational modification, where like you phosphorylate a protein, you can serotonylate a protein. You can actually monoaminylate protein. So serotonin, dopamine, and norepinephrine themselves can be added as tags to proteins, directly regulating protein function. A very cool paper from Ian Mays's group at Mount Sinai shows that if you serotonylate histones, you directly regulate transcription. So this is a receptor-independent action of serotonin, where serotonin goes into the nucleus, where it is added on by transglutaminases onto specific histone residues. And then associated with that, there is the recruitment of transcriptional machinery that can drive and promote transcription in most cases. So this is important to know, because I think, especially in psychiatry and neuropsychopharmacology, the non-receptor mediated, non-canonical or non-prototypical actions of serotonin and the monoamines are underappreciated. And they may become actually really relevant to think about, because keep in mind that for at least several billions of years, serotonin and other monoamines were doing this without any receptors in play. So there's a long evolutionary history for these roles to have evolved independent of the standard, more receptor-mediated effect on neurons that we have spent a preponderance of our time studying. It's important to keep this in mind. I like this particular review article from Ifran Asmintia because it's very provocative. The title of this review article is something like Serotonin, Sunlight, and Suicide. A very provocative review article, but the article is interesting because what he's talking about is the earliest stages of trapping light on Earth, which is the first step through which life evolved, because you have to be able to trap light to be able to generate food, and eventually that allows plants to make food, which is then, that's how all of this began. Life on Earth, as we understand it, began. But as a byproduct of this, his argument is that chromophores that were associated with trapping life, many of them contain tryptophan. And tryptophan is just an amino acid that happens to trap light. And so because of that, you have a way by which those chromophores can use light to generate, eventually through photosynthesis centers, allow you to generate energy. But a side effect of that and a byproduct of that is you produce free radicals. And associated with that, you have to come up at the same time with an evolutionary plan to be able to quench free radicals. And so antioxidants would have had to co-evolve along the side with the ability to do photosynthesis. And his argument is that which is the tryptophan-derived moiety that might have played a role at that point in time, and the tryptophan-derived moiety he speculates is serotonin. That serotonin may have co-evolved at the very earliest stages to work as an antioxidant to quench free radicals associated with the gene, and serotonin is a potent antioxidant in plants, that's for sure. Body of literature, which I would certainly recommend to anyone who's interested in serotonin, and many people I'm presuming in this audience are interested in serotonin, to read about the known literature of serotonin in plants. It does pretty much everything, right? From germination to modulating stress responses to, so there's all this body of work there where serotonin has these interesting effects in plants which are not canonical receptor mediated. So there is this work that is out there which is a little underappreciated in the neuropsychopharmacology community. Okay, so we stumbled on this observation where we found that when you put serotonin on neurons, you ended up with more mitochondria. That was the observation we stumbled upon. And now I'll tell you what we actually found. It makes now intuitive sense that it would have done that, but that is intuitive sense in hindsight. Everything is always 20/20 hindsight. At that point in time, we had not expected it. In fact, we found it very hard to believe in the beginning. We kept repeating the experiment to be absolutely certain that this is indeed what we were seeing. Now it makes sense because serotonin is a regulator of neuronal plasticity, growth, differentiation, aging, mood, etc. Mitochondria are central players in most of these processes. So in 20/20 hindsight, it seems very intuitively obvious. And if you had read the literature for plants, which we had not, we might have actually stumbled on it a lot earlier, simply that we just hadn't been looking. Just ignored that component of literature, unfortunately. So here's the question. Does 5-HT regulate mitochondria? If it does so, what aspect of mitochondrial function of biogenesis does it regulate? Is this important? Is this even relevant? And how do we begin to study this? So this is the team whose work I'm going to be talking about. The person who's led most of this work in the lab is Sashena. Sashena was formerly a graduate student, postdoctoral fellow in my group, and then now she's a research assistant professor who closely works with my team. So I'll walk you through the data, and I guess we'll just take questions at the end, but hopefully if there's something that needs to be clarified, you can just ask. All right, so let's just dive in. What Sashena did was she was growing cortical neurons in a dish in culture, derived from embryonic 18.5 cortex. And when you just grow these neurons in a dish and bath apply serotonin, and I'm just telling you that concentration is in the range of what you would expect at a synapse, because there's about 6 millimolar within vesicles. Okay, that's the sort of concentration you're talking about, very high concentrations within vesicles you'd be expecting in the micromolar and nanomolar range as you diffuse away from a synapse. You bath apply serotonin, and you get this increase in mitochondrial DNA content. And you also see this increase in ATP. This is cellular ATP. That is a measure of sort of ATP production, not just from OCR, which is oxygen consumption and oxidative phosphorylation, but also from glycolysis. So it's a cumulative measure of how much cellular ATP you have in your neurons in culture, and that also goes up with serotonin. More qualitative measures of mitochondrial mass, which is to look at mitotracker staining, for example. So mitotracker staining, when you label neurons that have had bath application of serotonin, and you simply look at a qualitative measure of pixel intensity of the staining of mitotracker, you see a clear increase in mitochondrial intensity. That's also seen with other immunohistochemical markers. Here is a voltage-dependent anion channel that is expressed in the mitochondrial membrane, and you ask whether the expression of that has changed, and you can see that that also goes up. Sort of more semi-quantitative approaches where you look at a transcription factor, TFAM, which is vital to the production of mitochondrial DNA and eventually mitochondrial transcription. That is also increased in response to serotonin. Cytochrome C goes up, VDAC goes up. And all of this cumulatively starts pointing to the fact that you have more mitochondria if neurons are seeing serotonin. I'll tell you that which neurons matter, because if you put cortical neurons, you get this effect. If you put hippocampal neurons, you don't see this effect, okay? So there is some region specificity to this, and that becomes very interesting in terms of whether this is a receptor independent or a receptor dependent effect, which I haven't told you yet, right? Both are possibilities. This is just showing that if you actually look at neurons in a dish and now measure oxidative phosphorylation, so this is now a readout of the functional readout eventually as a consequence of having more mitochondria. Basal respiration goes up, ATP coupled respiration goes up, maximal respiration goes up, and spare respiratory capacity goes up. The way you do this is by using different substrates and inhibitors that allow you to break down components of the electron transport chain. So you can ask what is happening to each phase in a sense of OCR that allows you to break this down. The one I find most interesting is spare respiratory capacity. And why am I saying that? And let's think about this in terms of layman terms. We know when you're driving your car and that yellow light starts coming up, telling you that the petrol is almost running out, it's a warning sign that you better fill up petrol, otherwise you might run on empty. So it's the last bit that you keep in your tank just to protect you from running out by suddenly as an emergency. Spare respiratory capacity is your extra available sort of energy output or energy currency that you keep in the tank in case you are faced with an additional stress. So if you increase your spare respiratory capacity, it gives you the chance to buffer way more oxidative stress or potential toxic challenges. What serotonin seems to be doing is driving up spare respiratory capacity. And that's interesting because this is a neurotransmitter we associate strongly with stress buffering or enhancing the capacity to buffer stress. So this is a cellular buffering of stress. In a sense, at a cellular level, you see this buffering type of response. Okay, but all this is in a dish. This is actually happen in the brain. So here is, in rats, we actually put a osmotic mini pump right here at the back. Let me just see if I can get this pointer. That is a laser pointer. So you just put in the osmotic mini pump and infuse serotonin straight into the cortex over a duration of 10 days. And then you take out the cortical tissue and you measure mitochondrial DNA content and ATP, and that indeed does go up. So this is exogenous bath application by infusing serotonin directly into the cortex. What about endogenous release of serotonin? You can do that chemogenetically by using biogenic mouse lines. So here, for example, is a PET1-Cre marker line. So this is PET1-Cre AI9 dual biogenic mouse line, where AI9 would be expressed in PET1-Cre positive neurons. What this tells you is we can drive the marker in serotonergic neurons of the raphe because PET1-Cre will label serotonergic neurons. And using the strategy, you can use a chemogenetic approach to drive a DREAD into these serotonergic neurons. And now the DREAD receptor, which is ectopically expressed in serotonin neurons, can be driven by an exogenous ligand, which is a DREAD ligand selective for DREADs. And when you put that in, you can cause serotonergic neurons to fire, which you can see here. And in response to giving this exogenous DREAD ligand for a duration of 10 days, you can drive up serotonin release as a consequence of driving up endogenous serotonin release. You drive up mitochondrial DNA, you drive up ATP, and you see a clear increase in several markers that are known to be indicators of mitochondrial biogenesis. Okay. If you now take out from those animals, isolate mitochondria from those animals, and then run seahorse on them to actually look at oxygen consumption within mitochondria from animals where there has been just vehicle control or a driving of endogenous serotonergic release, what's instantaneously obvious is you're driving up state 2 and state 3 respiration and ATP production rate. Because this is normalized for the amount of mitochondrial protein, this is also telling you that you're driving up efficiency. Because there's two ways to interpret it. Serotonin increases the number of mitochondria. If the number of mitochondria go up, you increase the amount of oxygen consumption and OXPHOS because you just have more mitochondria to do the work. Alternatively, you also increase the efficiency of mitochondria. Our evidence points to the fact that not only do you end up with more mitochondria, but per mitochondria, your efficiency is going up. Okay, so that's what it's reflecting. All right. Now, is this one of the, in our world, the expected receptor-dependent effects of serotonin, or is this some absolutely non-receptor-dependent effect of serotonin that we happen to have stumbled on, where there's a role for serotonylation of some important pathway protein, which then drives all these changes? So that was the next question. In cortical neurons, pretty much every neuron is in the cortex is expressing most of the serotonergic receptors. So it's all 14 of them. So, you know, this is going to be a difficult one to tease apart. Turns out, and very luckily for me, turned out to be my favorite serotonergic receptor, which is the 5-HT2A receptor. And that's turned out to just be fluke and interesting. That does not preclude the role for other serotonin receptors. And they're certainly well worth studying. But for the moment, I'll tell you that it turns out it's a 2A receptor. So if you bath apply serotonin, but you don't allow it to work on the 5-HT2A receptor using a drug called MDL-100907, which is an antagonist of the 5-HT2A receptor, you completely lose all these mitochondrial effects of serotonin. And that's true with mtDNA, that's true with ATP, that's true with multiple measures that we have tested. So you need the 5-HT2A receptor to mediate these actions of serotonin on mitochondria. And that's also true using a genetic strategy, where here using mice that have a very interesting knockout strategy. In these mice, they don't have, so the 5-HT2A null doesn't have the 5-HT2A receptor, but it has a neo-stop cassette, which is floxed. And that neo-stop cassette, because it is flocked, it can be removed using a recombinase. So you can put back the receptor. Okay, so it has a rescuable strategy. So here, for example, is the expression in the wild type. Here's the knockout. The knockout doesn't have expression. It's down to rudimentary levels. But when you start putting back the receptor by floxing out the rescued, so you're rescuing it by putting back the, removing the neo-stop cassette, you can start restoring expression. And when you do that, I'll just walk you through this part of the slide. We'll ignore the rest for now. Here's the effect of serotonin in wild type neurons. In knockouts, you get no effect on mitochondrial DNA. So you don't have the receptor. You will not see these effects of serotonin. But you start putting back the receptor by restoring it, the effects come back. Telling you that this receptor is central to these mitochondrial effects of serotonin in this circuit, at least at these times. Okay. If this is then independent of serotonin, if you simply stimulate this receptor, you should be able to reproduce this effect. The reason this receptor is so interesting is it is the target of many, many interesting drugs, including the serotonergic psychedelics. So the psychedelics, most of them will hit the 5-HT2A receptor with very high affinity. In fact, their tendency, their hallucinogenic potency is strongly linked to their affinity for the 5-HT2A receptor. And the 2A receptor is a vital component of some of the subjective effects or the psychedelic-like effects of these drugs. So here's a particularly potent serotonergic psychedelic, DOI, which is a phenylethylamine, which hits the 5-HT2A receptor. Just to be careful, it's not just a 2A receptor ligand, also hits the 2B, also hits the 2C. So be aware that it hits more than just the 2A, but it does hit the 2A with very high affinity. If you stimulate this receptor with you use DOI, you stimulate this receptor, you can phenocopy the effects of serotonin. So like serotonin, simply stimulating the 2A receptor is also enough to give you this increase in mitochondrial DNA and ATP. And similarly, it drives up OXPHOS. We don't have to worry about the details here. The bottom line is what you see with serotonin, you also see with DOI, except that you see it much faster with the psychedelics. Here, I'm showing you timelines, which are 3 days. I showed you the serotonin timelines. Those were like 1 week. You can also see it within 3 days, but the serotonin effects are not within hours. The effects with the psychedelics are within hours. We are seeing these within 2 hours, within 4 hours, and cortical neurons in a dish, also in vivo in the brain. And that opens up really interesting ideas because what happens with SSRIs, what happens with serotonin, broad modulation of serotonin versus when you targetedly go after the 5-HT2A receptor. This is all the in vivo data. In vivo data, you've given rats DOI every day, single injection for 3 days. You take out the cortex, you see this boost in mitochondrial DNA. Several markers, so this is now transcriptional expression of several markers that are associated with mitochondrial biogenesis. They go up. If you isolate mitochondria and look at seahorse, you can clearly see that you're driving up oxygen consumption, OXPHOS is up. And that's also seen with reference to multiple markers in Westerns, where you can see that players that are associated with mitochondrial biogenesis or function are going up when you stimulate the 5-HT2A receptor. For something? No? All right. So it's a receptor effect, is this particular, right? What's downstream? I'm going to skip this part. In the interest of time, I'm actually just going to tell you what it is, and then I'm not necessarily going to spend all the time showing you the data. Let's just look at the pathway, okay? Turns out, in the brain, you're using similar pathways for metabolic modulation of mitochondria as you use in your liver, as you use in your muscles. So there is a very well-known canonical set of pathways that drive up mitochondria. It includes PGC1 alpha, and SIRT1, which are major master modulators of mitochondria that drive up a transcriptional program in the nucleus that includes TFAM, NRF1, NRF2, which then get transported and out of the nucleus into mitochondria to drive up mitochondrial transcription, to drive up mitochondrial mass. This is how you do it in the liver. This is how you do it in the muscle. When we were starting to look at this in the brain, there was no reason to imagine the brain might do something drastically different. It doesn't seem to be, but it doesn't preclude the possibility that many other things are happening. What we seem to have stumbled upon is that in the brain, one of the drivers is monoamines. The pathway seems to be this conserved pathway, very well understood conserved pathway, extensively studied in the liver and in the muscle. But in the brain, serotonin seems to be one of the upstream regulators of this pathway through the 5-HT2A receptor. And if you now block SIRT1 signaling, so it's a histone deacetylase, it's a broad spectrum deacylase. It deacylases many, many different proteins, and it is one of the hub metabolic sensors in a cell. If you block the catalytic activity of SIRT1, you actually block all the mitochondrial effects of serotonin. You also do the same thing if you block PGC1 alpha, either using a catalytic inhibitor or using a genetic knockout strategy. Here's just the genetic knockout strategy with SIRT1. You need SIRT1 and you need PGC1 alpha. Okay, so now what we have is that in cortical neurons, SIRT1 PGC1 alpha dependent pathway is driving up mitochondrial biogenesis, the expression of several biogenesis related genes, which increases the mitochondrial mass, drives up ATP, drives up spare respiratory capacity. The question is, of course, what is happening to ROS? What are the functional consequences? What does all this mean, right? In a sense, we're moving into the meaning stage of, okay, you increase mitochondria. What is it doing for a neuron? Having more mitochondria, what does this eventually mean? What are the functional consequences? In our naive understanding of mitochondria, we thought that if you drive up mitochondrial mass and you increase the number of mitochondria, associated with this, you probably drive up ROS because you're driving up OCR, and a byproduct of this should be free radical production. That just seemed an intuitive understanding. Of course, remember, all the literature on plants points to the fact that serotonin can quench free radicals. And if you look at just this data here, now we're looking at simple DCFDA staining in cortical neurons in a dish when you have a bath-applied serotonin and challenged with either an excitotoxic substance like kainate or challenged with bleach, which is essentially oxidative stress. So what you see is very interesting. As expected, kainate and H2O2, which is because they're toxins, they will drive up ROS very strongly. But look at what happens with serotonin by itself. If you simply bath-applied serotonin to cortical neurons, you're scavenging ROS baseline. And if you've had a prior exposure like this, and then you come in with an excitotoxic challenge, you will buffer that challenge. In fact, you buffer that challenge quite strongly. And that makes sense from the perspective of the effects on spare respiratory capacity, from the point of view that you are, in a sense, endowing these neurons to actually buffer cellular stress quite well in the presence of serotonin. That might be because serotonin drives up SOD2 and catalase very, very strongly in cortical neurons. You get this very big transcriptional increase in SOD2 and catalase, but we haven't pursued this well enough. So we don't yet know. The jury's out on whether it's because of scavenging machinery that these effects happen or what else is actually going on. I'll just show you this graph on the right. This is a simple assay. We are just looking at MTT, cell viability in a dish. As we challenge cortical neurons with kainate, as expected, as you increase the concentration of kainate, neurons die. This is what you anticipate when an excitotoxin is put on neurons in a dish. But if you have serotonin pre-treatment, you will buffer an excitotoxin across a very wide concentration just by having had a prior exposure to serotonin. And for these effects, you need sirtuin signaling, because if you block sirtuin signaling, you will lose these effects. Now that opens up the possibility of a role for mitochondria. It doesn't nail it down. Opens up that possibility that you need it for these effects. I'm going to skip this part. The same thing, by the way, happens with serotonergic psychedelics. So that's interesting. This opens up questions about whether there is a role for this in conditions where serotonin is modulated. Obviously, serotonin, SSRIs directly target the serotonin transporter. You would be increasing levels of serotonin. In the process of aging, serotonin levels go down. Mitochondrial dysfunction also emerges associated with aging. Is there a correlation and a relationship between these two? Today, I'm actually going to tell you about the aging part, but I'll just touch upon what we are finding with the SSRIs, because that's still stuff that's not published yet, but it is a very exciting story. As expected, if you give an SSRI to an adult rat for 21 days or a little bit longer, 28 days, you drive up mitochondria in the cortex. That's not surprising because it would drive up 5-HT. As we have just seen, serotonin is a direct regulator of mitochondrial biogenesis. But it's all timing. It depends on when you give the SSRI. If you give the SSRI to a baby brain, and this is all rat, so just be aware it's preclinical data. If you give an SSRI to a postnatal, early postnatal so from postnatal day 2 to postnatal day 21, if you give it in that window, you actually have very long-lasting reduction in mitochondrial DNA, reduction in ATP, and a down regulation of OCR. So you end up, in fact, with lifelong changes in mitochondrial status that last out till 18 months of age. So these are very, very long-lasting. If you give it in the juvenile window, you get the exact opposite effect. You now increase mitochondrial DNA and you increase OCR and you drive up this sort of function, and that lasts again out till 18 months. So we very long lasting effects. You give it in adulthood, it's only on board while the drug is on board, all gone once you wash off the drug. So there is a huge amount of dependence on the age of the brain when you're coming in with serotonin and you're modulating serotonin. So even when we say serotonin is a regulator of cortical mitochondrial biogenesis, we will have to caveat it with timing, because everything I showed you so far was adult. And in the adult, it's a robust upregulate of mitochondrial biogenesis. But in the adolescent, it may prime the system for long term. Because in the juveniles, when we give the treatment from postnatal day 28 onwards till about postnatal day 48, and then look months and months later, 6 months later, we've just driven up function. And it's long lasting. It's not like what happens when you give SSRIs in the adult. So this has already begun to point out that there's going to be heterogeneity of response. Timing is going to be critical. Which are the receptors that are involved? I showed you the 2A receptors central in adulthood. I would not right now say anything about which receptor at the other ages. We have no clue, right? It might be other receptors, because all that I've shown you right now largely applies to the adult brain. But it's opening up the question that you have a very variant receptor milieu in the postnatal baby brain and in the adolescent brain. The entire milieu of 2A receptor function is, first of all, completely different in a baby brain and in an adolescent. You have much larger 2A drive in a young window, and it switches to 1A as it, especially in the neocortex. So that being the case, the balance is shifting. Signaling pathways are likely to be different. It opens up all kinds of interesting questions, but I'll show you a little bit of the data with the psychedelics, and then I'll show you the data in the aging context. It's interesting to think about the fact that the psychedelics are targeting mitochondria, and they're targeting them fast. It is much faster than anything you see with the SSRIs. It's much faster than if you went in just with serotonin, either endogenous or exogenous. It's a really rapid effect. We see it with 3 different phenylethylamine ligands. These are the 3 that we can easily work with in India. DOI, TCB2, and NBOM, all very potent serotonergic psychedelics, but none of them Schedule 1, so we can get them. We're in the midst of doing experiments with Viju, where we would love to do this with psilocybin and LSD to see whether this will hold true for the psychedelics that are in clinical usage, because this would be really interesting to look at. Here's the data. Here's the early time point data. This is 2 hours and 6 hours with DOI. 2 hours, you already see a really robust effect on mitochondrial DNA. This is in culture. So when you treat neurons in culture with either DOI, TCB2, or NBOM, all of them will drive up mitochondrial DNA. Here is lyseride. Lyseride is a non-hallucinogenic ligand of the 5-HT2A receptor. So we were just wanting to check, is the hallucinogenic component even relevant or important? In this particular case, what you can clearly see is that at least at the, did I lose that? Is it coming back? Yeah. So here you can see that at a 2-hour time point, lyseride is doing nothing. Okay, I'll show you that it's not that it's not doing anything, it's about timing. Okay. Here's DOI TCB2 in males and females in animals. So this is the data that was in vitro. This is the data in vivo. Doesn't matter which psychedelic you're looking at, they're having very rapid and fast effects. That's true in both males and females. This is effects on ATP. But I want to show you this. If you come in with glyceride for 3 days, you will get an effect. But you need to do, it's a slower effect. So serotonin, non-hallucinogenic agonists are giving you effects. They're just slower. Whereas there's something going on with the psychedelics that changes the scale and speed with which you're seeing the response. You do see a much, much faster response with the psychedelics. And that opens up very interesting questions about the signaling cascades downstream. This is all 2A receptor dependent. You have a knockout of the 2A receptor. The effects of DOI and TCB2 are gone. So this is 2A receptor dependent because the psychedelics are slightly dirty. They hit the 2A, the 2B, and the 2C. They also hit some of the other receptors, but we know it's 2A dependent. You block it pharmacologically, you knock out the 2A receptor, you lose these effects. So it's a 2A dependent phenomenon. There's so many questions here, which will spend some time chasing. We're very interested in asking what's going on downstream of the serotonergic psychedelics that drives this. Is this important for the behavioral effects of these drugs? They work faster. They give you much faster effects on behavior. Within 24 hours, you get a potent antidepressant-like behavioral effect in preclinical models with drugs like DOI, TCB2, NBOM, psilocybin, LSD. This opens up, these are very fast effects. Is there a relevance to some of these fast effects eventually on behavior? We'd like to test that. All of these drugs evoke really rapid effects on architecture and structure, on spine density, on morphology within the medial prefrontal cortex. Are mitochondria players in driving some of this neuronal plasticity and neuroprotection? Is there a role for these psychedelics in driving stress buffering through ER mitochondria interchange? And we're interested in novel plant-derived psychoactives and their mitochondrial effects using remember we started with, that's how I started my talk, that we started with plant-derived substances, but that was not inspired by any logic. It was based on let's try these plants. Now we know the structures that might be having effects. It's easy to go back and look for plants that have structures that structurally overlap with the indole alkaloids or the isoquinoline alkaloids. By the way, all the psychedelics, many of them start with their inspiration being plant-derived moieties. They're all potently plant-derived. Psilocybin itself is from mushrooms, right? So many of these plants have a clear derivation from, many of these substances have a direct derivation from plants. So we're interested in doing that as well. And one of the examples of this is to go after molecules within Indian traditional medicinal literature. Berberis vulgaris, berberine being a very interesting example. It has an isoquinoline alkaloid. That's the molecule berberine. And when we went in with berberine, inspired by the fact that the structures open up the possibility that it is hitting these pathways, we get very interesting effects on mitochondria. They are slower than what we see with the serotonergic psychedelics. They don't give us behaviors that we associate with classical hallucinogenic-like responses, but they have potent effects on behavior and as far as mood-related and despair-like behavior is concerned in preclinical models. They've also been used in Indian traditional medicine with some sort of reports of anecdotal and empirical evidence that they might have mood-related effects. But these are interesting molecules nevertheless. What's interesting is the 2A receptors getting targeted in some way, and we're very interested in looking at this further. This is work done by Sashena in collaboration with my dad, Dr. Ashok Vaidya. I'll just skip the fact that the pathways seem to be similar. So there's lots here to do and lots more unanswered than we have answered, but I think we've stumbled on something super interesting, that there might be this old conserved function of serotonin, maybe of other monoamines as well, which involves an effect on bioenergetics. And bioenergetics are central to neuronal survival. They are central to neuronal function. And by modulating such a central pathway, serotonin may be ideally placed to drive up stress buffering in critical limbic brain regions and circuits. The circuit dependence of this is also very, very interesting. Why PFC, why MPFC so much over other circuits? Maybe receptor distribution, etc. Let me show you some of the SSRI data. There's a little bit, I stuck this in here saying I can't give a talk at NIMHANS and not have an SSRI in my slides. So here's the one little bit of data that I have with SSRIs. This is in old animals. So now these are 13-month-old middle-aged rats. When you put them on fluoxetine in the drinking water for a period of about 3 weeks, what you can see is you drive up OCR. I'm just showing you the OCR data. So this is mitochondrial function. Now, why is this interesting? By 13 months, mitochondrial function is now already in pretty steep decline. It's telling you that there is buffering room here to keep driving it back up. There's still room left in the system to drive this up. So you get these effects. Timing is quite critical. And here is even older. This is 18 to 20-month-old mice in which we are driving up endogenous serotonin release by driving up mitochondria, by driving up the PET1 neurons. And in these neurons, again, you drive up a robust effect on cellular ATP and also drive up OCR. I just want to touch upon this because it tells you that will open up interesting other targets potentially for things that modulate monoamines, neurodegenerative pathways being potentially of relevance and interest here, though that is not usually the place that you think about SSRIs. Okay. I'll wrap by just showing you one last piece of data. I'm going to just show you the summary that when we found this effect with serotonin, we also looked at norepinephrine, we also looked at dopamine, we also looked at neuronal activity in cortical neurons. Neuronal activity will not do it, dopamine will not do it, norepinephrine will not do it in cortical neurons. And I had a very talented graduate student who said, how can norepinephrine not do this? We have to look at this better, right? So she decides she's going to look at it, switches circuits, goes to the hippocampus. In the hippocampus, serotonin does nothing, dopamine does nothing. Turns out norepinephrine is a big regulator of mitochondria in the context of the hippocampus. And that's all Darshana's PhD work. I'm not going to show you the data, but the bottom line is it is mediated via norepinephrine through the beta-2 adrenoceptor, which through a cyclic AMP PK cascade via PGC1 alpha mechanism drives up mitochondrial biogenesis in this circuit. Work that is emerging now from Mary Lobo and Carmen Sandy's lab points to the fact that dopamine may do this in the nucleus accumbens and the striate, which opens up a really interesting thought that you have monoamines that are distributing this work in a circuit-dependent manner, where you have different monoaminergic pathways doing this in different places. Now, this is not that hard to imagine because norepinephrine does this in cardiac myocytes. So this is known. That mitochondria are regulated by norepinephrine in the cardiac tissue has been known for a while. Independently, there's also an interesting other series of papers which are super interesting where when you burn fat, in a sense, thermogenesis is essentially also driven through beta-3 adrenergic receptors in fat cells. And that's through a mitochondrial modulation of the uncoupling proteins that allows you to burn up that fat, right? So that's again a mitochondrial pathway, different in the manner in which it's being hit, a different receptor. But this is opening up the interesting possibility that these really evolutionarily old neurotransmitters may have functions that precede their classical roles as neurotransmitters, where they might be doing interesting things to bioenergetics per se. Okay, I'm just going to skip all this. This is just all that data. Just want to, here's my one clinical atomoxetine in the clinic for treatment of ADHD, what it's called in India. No, but atomoxetine is used. And atomoxetine, because it drives up norepinephrine through a beta-2 adrenergic receptor effect, robust regulation in vivo in preclinical studies of mitochondria, telling you that something interesting is going on here. I'll skip this in the interest of time. So I'll end with this slide, opening up the idea that sometimes you stumble on interesting observations if you just play around a little bit. And this is an example of Sashena deciding that she wanted to dump on some serotonin and look at mitochondria. It changed the trajectory for all of us over the last several years, because if she hadn't done that experiment just for fun and just said, I'll use this as a potential control, and then we ended up actually chasing this as something really, really interesting, opening up the idea that there are non-traditional targets for monoamines that we may not have yet anticipated that have real relevance. Mitochondrial pathways have shown up in most studies as potential players for the pathophysiology of a variety of mood and neurodegenerative disorders. This opens up really interesting targets for these molecules in that sense as well. And I think that's it. Let me acknowledge the people. Most of the work I talked about today has been led by Sashena. And then I didn't get to talk about Darshana's work, but Darshana was like, there cannot be no role for norepinephrine. We just haven't looked at it well enough. And sure enough, in the hippocampus, there is. people in the lab, and here's the team. He's a mitochondrial physiologist at TIFR. It's wonderful to have not a mitochondria person. We stumbled into this in a sense. I've had the rare privilege of working with the approach at molecules that might come from, there are very interesting molecules in there because there are things that produce hallucinations that plant use in Indian traditional medicine, evidence of hallucinations being driven. Just don't know if it's to receptor mediated, don't know if it's a psychedelic-like substance. It'd be interesting to look at. Dr. Sheetal Suryavanshi, animal core at TIFR. Without him, we wouldn't be able to do much of our work at TIFR, and I can stop questions. How do you want me to do this? Just take questions? Yeah.
So I have two questions. So what triggers serotonin? Maybe I'm feeling like, when we pedal a bicycle or heavy lift, so there will be a drastic production of tryptophan. Ultimately, that would trigger the serotonin, right? That my question again, that should keep us more happy at some point.
It's very circuit dependent eventually, all these roles. I would not go, stress is a big regulator of serotonin itself. So when you are under conditions of stress, even if it's like a tail pinch in a rodent, you'll see a super big drive up for serotonin immediately associated with an acute stress. In fact, with chronic stresses over a period of time, you deplete serotonin. So that's what tends to happen. With an SSRI, in the first few, actually in the short duration with an SSRI, you reduce serotonin because you cause inhibition through the 5-HT, auto-inhibition through 5-HT1A, you actually reduce RAF-A burst firing. So transiently, soon after the SSRI comes on board, you have this window of a few days until you downregulate that 1A response drive. You will have a dip in serotonin and then it comes back. I'm sure the clinicians would know this potential room for angiogenesis in that first window when you are with the SSRIs over time. I think in popular parlance, the tendency tends to be to view an enhancement of serotonin as associated automatically with an improvement in mood. That's absolutely not true because in each circuit, it will be highly dependent on which receptor you tackle. So, if you go in, for example, in the amygdala with the 5-HT2C, serotonin, there is a robust driver of fear responses. So it's complicated. So I would not want to give a straightforward answer to that. I think the question you're asking, or maybe let me rephrase it, is that baseline, our brain is bathed with serotonin at most times. Unless you are like really depleted for some reason, there is actually a fair amount of volume transmission of serotonin going on. So I think that actually might be maintaining some degree of bioenergetic tone.
Then that's okay. Then what about the excess of tryptophan?
Excess of tryptophan doesn't automatically result in excess of serotonin. Depletion of tryptophan does a better job of reducing serotonin than the other way around. So tryptophan-rich diet does not automatically drive up excess serotonin. Excess serotonin will be the kind when you get a serotonin syndrome, which you can do with like a combination of TCP, L-tryptophan or something like that, where you block, catabolism of serotonin, you drive up the, production. But that, I don't know if clinically one sees that sort of excess of, I have no idea. I'm not a clinician, so I don't know how to answer that, but.
Or the melatonin is independent.
So melatonin in this circuit, at least for these effects, doesn't do it. There is now evidence that melatonin may regulate mitochondria in the pineal. So that opens up, this is now opening up the possibility that each monoamine seems to have done like a distribution of workload. So very speculative right now and highly hand-wavy because the data is yet missing for most of the monoamines. Our work shows that serotonin does this with mitochondria and the cortex. There's work from Rick Schnellman's lab that shows that serotonin does something similar in the kidney. So that's known. There's opening up a work now that serotonin does something similar in the gut, but it may be different pathways. So there's that data. There's a lot of data with cardiac myocytes showing clear effects of norepinephrine. Our work is unpublished, but we are showing that there's a big effect of norepinephrine through the beta-2 in the hippocampus. Carmen Sandy and Mary Lobo's work suggests that it's D1 receptors in the striatum and in the accumbens, but the pathways are not yet understood. There's some work that indicates melatonin in the pineal. All of this broadly suggests that monoamines are talking to a signaling cascade that downstream of which is bioenergetics via either enhancing mitochondrial biogenesis or driving up eventually efficiency. We have a whole missing link on looking at mitophagy. We haven't even touched that. There isn't evidence yet for it. It's something well worth looking at. But I think we'll stumble on more. Now, I think there are enough people interested and enough people looking from with different monoamines, different circuits. I think this will be heavily circuit dependent. It will depend on the circuit. It will depend on the receptor and the coupling of that pathway. Because we have bath applied serotonin for long durations in hippocampus. We see absolutely nothing. So it is circuit dependent.
Thanks, Vidita.
Sure.
Always pleasure.
Yeah. Thanks.
Hello, ma'am. So my question is that it seems that it's counterintuitive that the number of mitochondria also drives up the efficiency. So how do you measure that and why do you think that is?
The number of So the number of mitochondria doesn't have to drive up the efficiency. You could just increase the number of mitochondria and so increase oxygen consumption simply because you have more mitochondria to do the job. That's not what we are seeing. What we are seeing is that if you normalize for protein content of mitochondria, so that tells you that you're normalizing for ETC equivalence in a sense, you still see an increase in efficiency. Now that either happens because you change the essential efficiency or coupling of the electron transport chain. How you do that, we don't have evidence on at the moment, but it points to an enhancement of efficiency, not the other mechanism, because you have more mitochondria, so you have more OCR. How, we don't really know yet. Now, it would point towards something happening within the ETC in terms of coupling.
Thank you for an insightful talk. My question is related to, like you said, serotonin could have non-scavenging or receptor-mediated antioxidant activity. So, what is your take on involvement of non-neuronal cells like glial cells, which also express IH2 on these?
Great question. You're telling me all the things that we are in the midst of doing right now. So I have a graduate student who's looking at it in astrocytes. Preliminary evidence, take it with a pinch of salt until we are absolutely certain no effects in astrocytes. 2A receptors are abundantly expressed also on astrocytes. We are not seeing it in astrocytes. So it is neuronal at the moment. So we haven't looked at microglia. We haven't looked at oligodendrocytes. So we don't know yet, but it points towards it being neuronal at the moment. In culture, anyway, we are at like 99% pure neuronal cultures, but in vivo, it could have been any of those cells. It doesn't look like it's astrocytes.
I'm going to read out some questions from the Zoom now. H3 asks, is the number of mitochondria increasing or mitochondrial density increasing upon 5-HT administration?
The number of mitochondria is increasing. It doesn't look like density necessarily. I mean, density will go up if you increase the number of mitochondria. The question is it distributed? We don't really know. From our cortical neuronal cultures, it looks like it's uniform across the cell body. The increase in density is uniform across the cell body and the neurites. It doesn't look like it's selective to only the cell body or only to the neurites. When you are looking at it with bulk biochemistry, you just see an increase in mitochondrial mass. You could do the same thing in many ways. I mean, the thing is, you could do this with fission, fusion. You could do this in local dynamics happening. We don't know about through live imaging exactly what's going on. But if you increase mitochondrial numbers, as we seem to have increased using all the biochemical measures, you're bound to have more mitochondria packed wherever they are. And right now, you see that with mitotracker staining, that we have more intensity, which points towards more density. But till we actually look at this using dynamics, I will hold out on saying exactly what's going
on. The next question is from Prajwal. What happens to neuronal mitochondrial morphology, levels of DRP1, OXA1, and mitofusion1 upon psychedelic treatment in vitro and in vivo?
Yeah, great question. Is still doing those experiments. So I don't have an answer to that. We'll have to live image. We don't seem to be changing DRP1, mitofusin, OPA1, any of these transcripts don't seem to be targeted. So it's not a transcriptional regulation at least, but that does not preclude the possibility of direct effects on fission-fusion dynamics. We just haven't looked. We went after the biochemical and functional approach first. We'll get to the imaging now.
Next question is from Nilekani. The heterogeneity that is seen in the juvenile phase, can you please shed some light on the potential implications of these real-life scenarios?
Okay, so the reason we started doing this is inspired very strongly by work that came out of Columbia. Mark Ansoke, Jay Gingrich, and multiple people at Columbia. This is actually inspired by clinical studies where there have been reports that childhood usage of SSRI is linked to enhanced suicidal ideation. So that idea has been there from the clinical literature for a while. That led to Gingrich and Ansoke doing experiments in preclinical models where they went in and treated with Prozac, so with the SSRI fluoxetine from the window postnatal day 2 to postnatal day 21. That would represent in terms of, and this is a difficult jump to make from the preclinical model to the, what age does P2 to P21 mean? People think of it as last trimester, first 2-3 years of life. That's sort of the neuronal circuit associated timeline that people, but this is difficult to make. So take it with a little bit of a pinch of salt. In that window, so from P2 to P21 in rodents, when you come in with, and this is rats and mice both, if you come in with an SSRI, you will program lifelong increases in anxiety-like behavior and despair-like behavior in animals that are seen out across the lifespan. We have replicated their work multiple times, so have many other people around the world. It's out till 18 months. They're really old animals just from that short duration exposure. We were very interested. That's a window in which, by the way, 2A function is higher than relatively 1A function. And serotonin tends to be excitatory in the neocortex in that window. As you move to the juvenile window, something interesting is going on. People have a big debate about what exactly is happening in the juvenile window. But our work and work from Carlos Bolanos' lab suggests that if you come in that window, now postnatal day 28 to postnatal day 48 is the equivalent of, I guess, in the window 14 to 22 years. That's the sort of broad window people are thinking about. In that window in preclinical rodent models, when you come in with an SSRI, you reduce anxiety-like behavior and you reduce despair-like behavior, again, for the rest of the lifespan, out till 18 months. So there are very long-lasting consequences of an SSRI in those two windows. You do the same thing in an adult or even in an old animal, all the effects are only when the drug is on board. You withdraw the drug, the behavioral effects go away. So this is what the preclinical data suggests. We don't have a good clinical correlate, obviously, for this because there isn't a good comparison, but the preclinical data is inspired by the clinical observations that there are very different effects depending on the time window in which the SSRIs are administered. This is also, by the way, fluoxetine hasn't been looked at extensively with other SSRIs. There's some evidence that citalopram gives a similar effect, but nobody has looked at it closely with other things. And certainly drugs like dizipramine in this window don't do this. So there's something going on with enhancing serotonin in these developmental windows that can have very, very long-lasting consequences. I don't know if that answers that sort of, yeah. Yeah, not on astrocytes of psychedelics, certainly doesn't look to be with serotonin either. We haven't looked with norepinephrine in the hippocampus yet.
Next question is, how are SSRI metabolized, especially how they cross the blood-brain barrier after being metabolized? And will SSRI stay lifelong in the adult human brain after 6 months?
No, they will certainly not stay. They're all washed out within a period of about a week. And people have looked at this quite closely, even in rodent preclinical models in the postnatal window, because one concern was what the heck, maybe they're completely differently metabolized the baby brain as they are. That doesn't appear to be the case. So downstream metabolic indicators are similar to what has been seen clinically. It's also similar to what is seen in adult models. It washes out. So you're not having a long-term effect of the drug just hanging on. It is possible that the drug fundamentally changes some of these metabolic pathways. That has not been looked at very carefully. So now that just hasn't been done. So no way of really knowing what happens to the P450 pathway, for example, in that early window.
Can serotonin be used as potential therapeutic to be used for old age people to restore mitochondrial efficiency?
super tempting. I don't know about serotonin, but I can tell you that there are in the pipeline from Beckley Foundation and many others, patents right now for psychedelics for neurodegeneration. So that's already in the pipeline. So that's not very surprising. I mean, it is 2A receptor dependent in that old age. We get the same effects with DOI. So that opens up. I certainly can imagine that people are going down that path with the psychedelics for sure. Psychedelics are being talked about for macular degeneration as well, retinal disorders as well. So it's opening up the multiple places in which people are looking at psychedelics as potential leads for. So neurodegeneration is certainly a candidate for at least the drug companies who are interested. So I know, for example, Beckley is, but yeah.
One basic question: does the mitochondria behave in different tissues in similar fashion, and how are they interrelated?
A great question. I'll point him to the paper that just came out from Martin Picard's lab talking about heterogeneity of mitochondria circuit by circuit in the brain. They are extremely heterogeneous, and we have poor understanding of the heterogeneity at the level of morphology, at the level of function. So there's heterogeneity. Absolutely. There's also a big question. I mean, we keep talking about energy currency and energy budget in the brain, saying that the brain is using 20% of most of all our energy consumption is being utilized. We have no idea about the circuit-specific allocation or demand of energy equivalents. That will vary, and we don't know that. And we also don't know that contextually, because that could change depending on context. So this is an open, big biochemistry question of what is the actual energy budget in a neurocircuit manner, because it's not going to be allocated identically, and that will vary contextually, strongly vary. I certainly would suggest that paper by Martin Picard, and there's also a very recent paper by Martin Picard, which is one where it's looking at whether trait personalities correlate strongly with certain mitochondrial markers, especially for the complex 1 of the ETC. And that they did in postmortem tissue where there was deep phenotypic profiling. And so that's actually an interesting paper that just came out in PNAS. Again, pointing to associative, correlative, but very, very interesting ideas being put out.
This is from Vidita. Why would you think the effect of 5-HT is circuit dependent? Do you think it's just the receptor expression or something else?
Currently, I think it's receptor expression because that's what I can easily estimate. The 2A is very abundantly present in the cortex, is barely there in the hippocampus. It's there on very important cells, but if you look at global expression, it's low. It's in largely inhibitory interneurons in the ventral and dorsal hippocampus, whereas it's on almost all projection neurons in the cortex and inhibitory neurons and astrocytes and microglia. So it's very, very abundant in the neocortex. I don't think it's just expression because it's also fairly reasonably expressed in the striatum. I don't think we're getting effects there. So it might be coupling of those receptors downstream to the signaling cascades. That's my guess. The hippocampus effects with norepinephrine in the cortex, there's all beta 1, beta 2, beta 3 are abundantly expressed, but we don't see anything in the cortex. So I don't have a good answer for this yet. Clearly, the receptors are a relevant component, but might not just be receptors. It's probably the recruitment of the downstream pathways.
The second question is, do you think there can be other ways, like sensory, to increase 5-HT that can have beneficial effect, at least in certain age brackets?
Really good question. I think there could be. Certainly, I think that so sensory, I'm going to go with a totally different potential lead to this. I would imagine that if you did fundamental regulation of arousal pathways in the brainstem, there might be an effect on most ascending monoaminergic circuits. And I'm talking about oxygen and pH control in the brainstem. And that then opens up very interesting possibilities of what this does with reference to breath control. It just opens up interesting ideas. That's all. There's no evidence of it, but it's sitting right there in the brainstem. The LC is right there. The raphe is right there. Everything, the ascending systems are right there. And all the pH and local sensing of arousal pathways as well as oxygenation is right there, right next to it. So it's tempting to speculate, no evidence so far. I'm sure there will be other things that regulate, can recruit monoamines and so regulate mitochondria.
Good talk. This is a basic question that you said the circulating modifies the resistance and have an influence on the transcription. And I think that in the neuroscience community, mostly focused on mitochondria, but ignoring too much on the nucleus. What is your view? Because that's not happening in the nucleus in terms of identification of collaborative regulation.
Yeah, I think that even the mitochondrial effects will eventually be nuclearly mediated because all the drivers are largely nuclear genes that then have to be transcribed, transported out into the mitochondria and then drive mitochondrial biogenesis. So I did talk to Ian Mays quite extensively on his sort of large scale chip. So he has antibodies that allow him to detect the serotinylation modification. And then, of course, he's sequenced everything that it's associated with to look to see which are the mitochondrial players that are potentially coming up. And there are some, he's obviously not mined the data from that perspective. It would be very interesting to mine the data from that angle because serotonin seems to, for the longest time, it was thought that it was an artifact that it was in the nucleus. That there was so much serotonin in the nucleus, people just kept thinking this is an artifact, it's cross-reacting with something, etc. But it is now, I think now the evidence is very, very clear that serotonin is serotonylating multiple target proteins. One of those are histones, but there are plenty of cytoskeletal proteins that are also serotonylated that have a variety of effects on cell function. So yeah, I think that you will have that also as a component. I wouldn't rule that possibility out. Right now, I'm saying it's a receptor-mediated role because we focused on the receptor-mediated roles. 2A is clearly a component of it. I wouldn't rule out that there might be an element that involves some serotonylation as well. We just haven't looked at it well enough. What we did do is we blocked the serotonin transport by blocking the serotonin transporters on these neurons, especially in cortical neurons. Many of them ectopically express the serotonin transporter. We blocked it. We still get the effects on mitochondria, telling us that the receptor is clearly a major player. I wouldn't rule out if there is some component that is also serotonylation.
Since you don't talk about CBM-1, which is sports, I guess, nuclear. There are many other situations which are
Yes. Yeah.
more specific mitochondria.
Yeah.
In your case, you specifically looked into the CBM-1.
Yeah, it could be 33, 4, 5. I mean, those are certainly candidate sirtuins. For the moment, our focus has been on 31 predominantly because that seems to be the major driver. It's like, it's a hub. So we are likely to see multiple other players also potentially involved. It's very, very possible. But yeah, we just haven't looked at it yet. Yeah.
Lovely talk. Just wanted to just check about one part about, you talked about the dose dependency with regards to serotonin. Is there a similar kind of dose dependency which is there with the psychedelics or with the SSRI?
Yeah, so SSRIs, we use doses of, usually we use for fluoxetine at 5 milligrams, 10 milligrams, and 15 milligrams per kilogram for a rodent, which is sort of based on what people have backtracked from clinical measurements using then plasma measurements, etc. So that's the dose range. Right now, the data I showed you is all 10 milligrams per kilogram data. We have not done, we've looked at 10 and 5. Both of them give you very similar effects. We have not done the juveniles with multiple doses. People have looked at 5 and 10 in the postnatal window. I don't think there's a massive dose dependence as far as the SSRIs, at least in the postnatal or in the adult window, seems to be the case. We also don't seem to see it either in terms of any inverted U, because we are hyper driving when we go in with a chemogenetic approach. Serotonin levels are really high. Even then, we continue to see it. So we're not hitting either a ceiling or actually the other end. We're not seeing an inverted U. The psychedelics are interesting. We're getting the effects with very low doses. Yeah, really low doses and very fast. So the effects are like sometimes 45 minutes to an hour even. So we are beginning to see very, very, especially on function, you can pick up the effects really, really quickly. So even relatively low doses, you're beginning to see these effects. Yeah. There might be a dose dependence certainly there.
questions, but we do have some tea and snacks outside, so you can definitely catch up with Professor Vidya and sorry.
That's fine.
And ask the questions. Thank you everyone for coming today. Thank you, Professor Vidya, for being with us today for this wonderful talk.
Thank you.
Have a round of applause for her.
Thank you.